good questions. And they can also move towards you. I'm just joking.
And I think we don't need the microphones, right? For the size of the room. I think people online.
Hur streamar du detta?
Ja. How was it?
I've seen men det er noe som er det hele tiden. Det er så kort med budget. Varför de blev inbjudna? Ja, jag tror inte det. Jag tror fordi at NY så var det
I think it's just modified the Octo approach to compare size files as well.
Sierras or?
I haven't heard of him in the past day.
Ekman.
More important stuff.
It would have been nice to have this email global data set.
Så det kommer så bli.
Ja.
I need the
16小时是2天，第二天第一天晚上16小时，第二天晚上再16个小时，我要开始调了，连睡3天我就不行了。
story. I'm surprised to get an email what to write in the story. I thought you would to read the paper.
就是我还想睡，但是睡不着了，所以就正常。
Kanske missförstått.
Okay.
So, today is a great day for the University of Bergen, for the Geophysical Institute and for the Nansen Center. We put to test the best we have, our young researchers and our research environment colleagues. So, it's a great honor and pleasure for me, on behalf of the university and the faculty, to welcome you all to this doctoral thesis disputation ceremony for Edson Filisbino Freire da Silva. He was admitted to the PhD program at the Faculty of Mathematics and Natural Sciences in November 2020. Time flies. And he has been supervised by senior researcher François Cournillon of the Nansen Center and researcher Julien Préjean of the Nansen Center and Professor Noel Keenley-Side of the Geophysical Institute, University of Bergen. Edson da Silva has been a PhD candidate at the Geophysical Institute, and on a big day, 1st of September 2023, Edson submitted his thesis, Prediction of Harmful Algae Blooms Impacting Shellfish Farms in Norway. And this for being evaluated for the degree of Philosophiae Doctor at the University of Bergen. The following committee was appointed to evaluate the thesis. Research Engineer, PhD, Raphaël Sauzet, Institut Le Maire de Villefranche, CNRS, and Principal Earth Observation Scientist, PhD, Peter Miller, Plymouth Marine Laboratory. And Professor Truls Johannessen of our own Geophysical Institute. And most importantly, the committee found the thesis worthy of being defended, hence we are here today, and the following opponents were appointed. Opponent, Research Engineer Raphaël Sauzet. Opponent, Principal Earth Observation Scientist Peter Miller. Members of the audience will have the opportunity to ask questions and present comments after the examination by the two opponents. And we now kick it off with Edson presenting his thesis. I have the honor. Take it away.
So, first of all, thank you for everyone coming here today to watch my PhD defense. And a special thanks to my opponents, not only for accepting to evaluate my PhD, but also to having all the work for coming from France, from UK, to be here today with us and to evaluate my case. So thank you very much. So I will start just by giving just one moment. That's okay. So first, just a summary of the contents, which I will talk about it. I will start by the introduction and then move to the objective of my thesis. Then I will have a very short description about the data methods, and then I will talk about the 3 papers that I had doing my work. And last, I will have the conclusion and some outlook for future work. So here in Norway, one of the most important shellfish are the blue mussels. The blue mussels, they are harvested in the industry sector. Nowadays, there's around 43 companies working on this field, which supplies the internal market in Norway and also exported to Europe. But it's a local tradition of the local people here to harvest the wild mussels that are in the coastal areas. So the blue mussels here is a very important food resource. So, talk about the biology of the mussels. I will not go into details, but what is most important here is the way that they feed on the water. So they are bivalves, they are filter-feeding organisms. So we have in the water column, we have some algae, some particles that goes in the inner shell, and then it's filtered on the gills of the mussels. Here in the gills, it's where the algae is accumulated, and then it goes to the digestive system, which makes the mussels grow. So, and one of the interesting parts about mussels is that they are very efficient filters. In the picture on the right here we have one example. So one aquarium without mussels, without bivalves, another one with some bivalves. Both of the beginner experience had the same quantity of algae and sediments on the water, and this is just after a few hours. So they are very efficient filters and can feed quite well on algae. So then I'll talk now about what is the problem that we can have with the mussels. And one of the main problems is the toxic algae. Some species or genus of algae, they can produce toxins. For example, Alexandrium species, Dinophysis, Azadinium, Protoceratium, and Pseudonychia. And those toxins are listed here based on the symptom or the name of the toxin, or based on the vector, that is shellfish. So for example, some species of Dinophysis, they produce diuretic shellfish toxins. Some of Alexandrium produce paralytic shellfish toxins. Azadinium produce azaspirate shellfish toxins. Protoceratium, iasotoxins. And last one, the Pseudonychia, can produce amnesic shellfish toxins. So as you can imagine, the problem that we have here, we have people that eat mussels. It's an important sector in the industry, but we can have some algae that can produce toxins, which can be fed by mussels. So we have 2 main problems here. First one, we have the health risk. People are poisoning if they consume those contaminated mussels with toxins, and depending on the concentrations can lead to death. And also we have the economic impact. For example, the shellfish farms are not allowed to sell the contaminated mussels because of the health risks, but then if the mussels continues contaminated for very long seasons, it can generate some loss of revenue. So we don't have the estimation of how large this impact here in Norway, but if you take, for example, Scotland, the closest region, only one of those toxins that I mentioned is responsible on average for reducing 15% of the gross sales profits. So for those reasons, we need some methods for dealing with this problem. So the most common method, and here in Norway, is done by the monitoring coordinated by the Norwegian Food Safety Authority, that is basically for preventing human poisoning. So there's 2 different approaches. Here it's a picture of one of the farms that we have here from the North Shell. And every week someone comes here and collects the water on the surface. This water is brought to the laboratory where the algae present here are identified, counted, and then it has its abundance estimated by cells per liter. If this abundance reach these hazard limits shown here in this table, for each toxic group of algae, the farms are not allowed to sell the shellfish. And usually, at least here in my thesis, when I refer to harmful algae blooms, I basically mean when the algae increases its abundance, reaching those levels. The second approach is monitoring the toxins in the mussels. Every month, mussels samples are collected in the farm and then estimate the concentration of toxins per flesh kilogram of mussels. So for example, for DST, we also have some hazard limits. That is 160 micrograms of toxin per kilogram of mussel. So this method works quite well here in Norway, and it's used mainly for the shellfish farms, but as I said, some people take mussels on the wild. So for this reason, people here in Norway are advised to take wild mussels close to those farms because we have data. So this data is also used for warning to the general public. So here it's one example. We have one region here that is in the Oslo region. We have the advice that is do not eat shells. When the samples were taken, the reason, that is for example Alexander is above the danger limit, and until when it's valid. And here we have the data for making this decision. And here we have the Alexander exceeding the hazard limit. So this is for avoiding human poisoning, but also the interest is to not only make safer, but also to help the industry sector. So for that, what we have that it has been growing recently, it's HAD predictions. So it's basically harmful algae predictions. Here I summarize in two different approaches. The first one is particle tracking models. So the idea behind is basically modeling the spread of a harmful algae bloom when it's detected. So if you take this picture, for example, here that is in the Norwegian coast, let's say a harmful algae bloom is detected here by in situ measurements or by satellite observations. Then it spreads on hydrodynamic models with the current, and then it has its spread modeling and to see if it can reach other farms in the region. This method is the most common method. It has been used already in Norway, but one limitation of this method, it relies on first detecting harmful algae bloom and then modeling the spread. A second approach that also has been growing, which I call here machine learning prediction. So the idea of this approach is to predict the harmful algae blooms before they happen. So the idea is to take time series of the data of variables that can trigger the harmful algae blooms. For example, temperature, salinity, nutrients, all those factors that combined can lead to the harmful blooms. And then it's fed into machine learning models that can be many different techniques. And those models, what they do is to learn the patterns of the environment that can trigger the harmful algae blooms and use this information for forecasting, which can be the forecast of the biotoxins on the mussel, or can be the forecast of the harmful algae in the farm. So, since there are machine learning, there are many different approaches and techniques of how to do this modeling. I'm not going into details of each individual method that we have published, but what I can summarize here is the current gap that we have in this approach. The first one regarding time series data, it's and the regional is that we don't have method that was developed to the Norwegian coastal waters using this approach. And the second reason is that few studies have addressed algae-related shellfish poisoning by using this approach. And one of the reasons is because for addressing those algae here, we need the algae identified at genus species level, which makes few samples because we are only relying on in situ samples. And then there's not many applications compared, for example, for fish farms that can use chlorophyll that can be helpful. So in my thesis, I address this gap that I'm showing here. So that is the objective of my thesis, that it was to calibrate machine learning prediction models of harmful algae blooms impacting shellfish farms in Norway. I follow this objective by three main ideas. So first one is to produce models ready to be employed and test. So I will not focus on only assessing what is causing the blooms, but also try to provide models that could be tested in a near future with the monitoring system or with the local farms. Second is to evaluate the skill of model sensing in model analysis in training those machine learning models for HAD prediction. And the main reason is because even if you have the algae data in the coast of specific toxic algae identified, we don't have data of the environmental factors. For example, temperature is not collected at the same points where the algae is monitored. So if we can use remote sensing and model analysis to produce models, we can use that basically to calibrate models anywhere where there is a monitoring system. And last one, the idea is to develop a calibration methodology that can be extended in other areas. And the main reason is because it's not only a problem in Norway, it's a problem elsewhere. So if you can produce a model that works here in Norway, would be interesting to expand that in the other regions. So all those three ideas here, it defines the methods that I use. So, so starting for the data that I use, I focus on using data that would be available worldwide, everywhere in the world. So for remote sensing, I have sea surface temperature, photosynthetic activity radiation, fire, chlorophyll A, suspended particulate matter, and northward and eastward winds. From other reanalysis, I use a mixed layer depth and sea surface salinity. But here the data that I use is from Topaz, which is only available in the North Atlantic, so for an extension would be only the North Atlantic, but there are some similar products available in other regions. And last, the data that I use is basically the algae abundance coming from the monitoring system in all shellfish farms since 2006. So basically the data that I mentioned in the introduction. So for the methods, for machine learning, I only used one method that is supported vector machine. But I have to say I made some initial tests during the beginning of my PhD, and SVM was the one that worked better. But also, what is the advantage of using SVM? The main advantage is because it works well with small data sets. For working with this kind of approach, we are relying on in situ data from the monitoring system. So it's few data compared, for example, compared with satellite data. There is a large volume of data. So if you're working with limited data, you want something that works well with limited data. And second, SVM can be used for many different approaches. Can be used for classification, can be used for probabilistic estimations, can be used for regression. So we can try and play with many different ways of calibrating, forecasting, and prediction models. And last, because SVM is very easy and straightforward to use and calibrate. So if you can calibrate here and it works, it will be easier to just extend it to other regions. So now I'll start to talk about the papers. So first I will explain what was the idea behind the paper's development. So the modeling of the algae-specific species can be too complex. So in the first paper, I focused on assessing the whole algae community. Basically, not algae, but phytoplankton, because it's also considered cyanobacteria. I did that by using the chlorophyll estimated from satellites. So the idea here was more simple, using satellite observation chlorophyll to estimate spring and summer blooms, and then simple inter-annual linear regression, that I want to see what would be behind the years that we have stronger spring blooms or later spring blooms, and so on. Then, by using these results, I moved to the paper 2, and now here I start to focus on specific taxa, as I mentioned earlier. So my target here, it was to estimate the probability detection of each individual taxa by using SVM, as I shown here in the scheme. So the idea is, based on the environmental conditions, what are the chances of detecting the harmful algae in the shellfish farm. And last one, in the paper 3, I increased the complexity of my target, and now I work with the forecast of toxic algae abundance, which is the variable monitoring the farms, for the Dinophysa acuminata, and I use a SVM regression. So the idea is a regression that takes environmental information and forecasts future algae in a subseasonal scale. So as I moved from less complex to more complex approach, I changed my study region. I narrowed down to a specific place. At the beginning, I focused on the North Sea, Norwegian Sea, and Barents Sea, here in this red box. Then I moved to the Norwegian coast for the shellfish farms that are along the coast, so inner fjords I had to remove. And the third approach, I worked only with one single farm in northern Norway. So at the end, I had 3 different papers. The first one, 21 years of phytoplankton bloom phenology in the Barents, Norwegian, and North Seas, was published in the Frontiers in Marine Science. Paper 2, probabilistic models for harmful algae, application on the Norwegian coast. It's now in review in the environmental data science. And the last one, forecasting harmful algae blooms, application to Dinophysa acuminata. It was published in the Harmful Algae Journal. So, starting for the first paper. So, briefly explaining, what are the spring and summer blooms here. So we have three important mechanisms here. During the winter, in these high latitude regions, we don't have enough light available, and we have a deep mixed layer depth. So the nutrients are accumulated here from the deep water to the surface waters. In the spring, we have the increase of the light. The increase of the light makes the phytoplankton start photosynthesis and growing, while the shallowing of the mixed layer depth makes the phytoplankton holds here in the surface. So that leads for what we call spring blooms. That is this half increase of phytoplankton biomass. Between summer, end of spring and summer, the blooms end because the nutrients are depleted, and also there's degrading to zooplankton. But then in the end of the summer and next autumn, mixed layer depth start to get deeper again, bring more nutrients, and we have this secondary bloom that we call summer blooms or autumn blooms, depending on the region. And then we have the light decreasing because it's autumn, we are going to the winter again, and then we don't have any bloom again until the next year. So here I'm focused on this system and looking to 21 years of data, looking if they are changing over the years, try to explain what is causing years of variation in the onset. So, but first I want to show this mechanism is not the same everywhere. So here I use a clustering method called K-means to cluster different regions where the seasonal time series behaves in different ways. As you can see, we have 16 regions. I will not talk for each region here to explain, but the point is, for example, sub-region 1, we have these two traditional peaks here in the Barents opening, while here close to the north of Scotland, we have a very long spring bloom and then a general decrease in chlorophyll. I just remember this is surface data because it's from remote sensing. So for each individual region, I looked into what was behind the changes or variability. So I have two main results. One is first for trends. And the first is that the summer bloom trends from 2020 and 2020. So here is the slope between this period. In the white areas, here for the onset, day and duration, it's where there is no significant correlation, no significant trend. Red areas, it's where we have a positive trend. And the values in the white boxes here, it's the value of the slope of the trend. So take, for example, here in the Norwegian shelf, here in the peak day, the trend is 1.23 days per year. So it means that in the whole period here, the summer blooms were delayed around 23 days, the peak day. So as you can see, the summer blooms here in the region, they are starting later, finishing later, and also becoming longer. The second main result, when now I look to the interannual variability, and here the main result is for the spring blooms, we have some interannual correlation with mixed layer depth and sea surface temperature. So red values, positive correlation, negative values, and the negative values are blue color. And the values of the correlations for the interannual variability are in the boxes here. So what we can see here, for example, for the mixed layer depth and intensity here, is that during the spring, when the mixed layer depth starts shallowing earlier, and it's shallow, the spring blooms not only start earlier, that in some regions they reach higher biomass in the peak. For sea surface temperature, we have some different and opposite relationships. Here in the open waters, this negative correlation is because sea surface temperature are changing the mixed layer depth, so that's why I have this kind of relation here. But we have an opposite relationship in the Norwegian coastal shelf. And the same we can see here for intensity. So what could explain why we have these different responses is that in the Norwegian continental shelf, we can have some trapped eddies bringing warmer waters, Atlantic warmer waters from the offshore region to the shelf. And this not only delays the bloom, but makes the bloom reach lower biomass. This result is published by another paper, but that could explain why we have this negative relationship again. For this region, in the north of Scotland, warmer temperatures could promote increased growth rate of phytoplankton, and that could facilitate the bloom reach higher biomass during the peak. So that could explain why we have a positive relationship. So now moving to the second paper, now I target specific taxa. So again, I had two different approaches here. One is present probability models. So basically the idea behind is using temperature, fire, salinity, and nuclear depth in the SVM model to estimate the probability of detecting at least one cell of each of this toxic algae here that we have here along the coast. This is the presence probabilistic models, and then there is also the harmful concentration models. The idea is the same, estimate the probability of detecting the harmful algae, but instead 1 cell per liter is the probability of reaching the hazard limits here, as we have in the table. So, okay, before I show the statistical results, I will explain about reliability diagram to understand how to understand if it's a good or it's a bad model. So basically, the reliability diagrams to show the correlation between probability and the observed frequency in the test data set, which is not the same as the one that was used for training. And that because probability is basically the observed frequency of an infinite number of trials. So there is 3 different features that we can consider here. Here, it's an example for Dinophis norvegica. So we want to this relationship to be close to the 1-by-1 line, because then the model can discriminate the targets that has lower or higher risks of detecting the algae. The second factor is that the model can predict the 100% probability. If it can, it says that the model can be confident in predicting the harmful algae, because it can give with 100% chance that the algae will be detected. And the last, if the model can if the model is able to predict the 0 probability, so the model can be confident in predicting no harmful algae. So take this example for Norvegica. What you can see, well, there is a line closed by 1-by-1, so it can discriminate regions or targets that are lower and higher risks. But as you can see, it cannot predict 100% probability. And the main reason is because we are using limited data. We are only using 4 variables, and those 4 variables is not enough for giving 100% chance. There are other factors, for example, nutrients, prey availability, that also controls the Dinophis norvegica variability in the ocean, but since it's not fed into the models cannot learn how to do that, how to forecast 100%. And it cannot also for Dinophis norvegica, the 0% probability. And that's because this is a common species detected almost along the year. Even in the winter, we can still have some species detecting on the farms, in the beginning or the end of the winter. So now, we look through the all taxa. So presence models, HC models. So for the presence models here, each color 1 different taxa. Not going to the details of each one individually, but what you can see, basically all of them will have this 1-by-1 line, except here, this pink color, which is the Reticulum, but still it seems to have some relationship. What we also can see is that none of those models can predict 100% probability because of the same reasons I already mentioned. But a few of the taxas, especially Alexandrium and Dinophis acuta, we can predict the values close to 0% probability. So we can almost entirely certainly, we can have huge certainties to say where there is it's not likely to be detected those harmful algae. But when we go to HC models, the results get worse. As you can see, it seems that now it's only working with 4 taxa, and the lines 1-by-1 is not that good as the presence model, but it's still significant here. While for a few taxa, for example, Pseudonychia, Dinophis acuminata, and others, we don't have any relationship. So having a better look to the statistical part, here for the correlation, for the plots that I showed. So here we have the correlation for presence modes. Here again, pink color, the correlation not that great, but it's above the significance level here, shown by the dashed line. While for the HC models, only half of the taxa are significantly correlated. So the question is why it's more challenging to HC models to provide some accurate estimations. So main reason we can see here in this plot. Here is the positive instance available for training. In the presence models, it means basically how many samples we have with at least 1 cell per liter. So that you can see we have many samples, but when you go to HC models, is how many samples we have with the hazard limit, above the hazard limit. And you can see there is a huge decrease in the data for training models. And especially in the ones that the models will show significant correlation. Which means we cannot calibrate models here because we don't have enough data still. The main reason. Right. So, but even though we can have some good presence models, a few for HC models, so how we can use that for helping, especially the farms and safeguarding public health. So here, one example of application to using the presence model for Tamains and Acuta. The models are applied every week for 1 year, and this is the annual average. So what you can see here that we can discriminate regions where it's more or less likely to detect harmful algae. So for example, Tamarense, we can see that the larger chances for detect them is in the northern Norway, and the reduced values close to 0 in southern Norway. But for Akuta, we have opposite pattern. It's more likely to detect here in the south than in northern Norway. So with this information, if someone planning to start a shellfish farm somewhere, they can use this information to decide. For example, if they go northern Norway, they will have to be more concerned about tamarins. Go southern Norway, not that much concern. And the same for Akuta and the other taxa that I'm not showing here. And what is also nice about using this model is you can predict where farms have not operated yet. For example, we have around 9 or 10 shellfish farms in Norway that has at least 10 years of time series. So the people there, they are already very knowledgeable of which harmful they should be concerned. But applying this model, we can extend to the entire Norwegian coast here. The second application is estimating periods of increased risk. So here, it's only one example for tamarense in Arendal, that is here in southern Norway, Vesterålen, northern Norway. So here, looking to the Arendal first, we apply to the week of the year. So what you can see here, blue color, presence model, orange color, HC model, and the red color is one harmful bloom that was detected. And again, this will apply to the test data set to see the viability of using it. So what you can see here, in general, most of the year, there's close to 0% chances of having a harmful bloom. But here, in this small season where the chances are increasing, it's when we detect a harmful bloom. And the same you can say to Vesterålen, but as in Vesterålen, you can see we have a very larger season when blooms can happen. So this information can be given to farms, it can be given to the monitoring system. For example, someone in Arendal, when it's between week 10 and 20, they could employ, let's say, more accurate methods, change the frequency of monitoring, and enhance the monitoring system. Just as one example. Okay, third paper now. So the first question here is why Danophysa acuminata and why northern Norway? So the first reason why Danophysa acuminata, we can look into this time series. So here we have the dark lines here. We have the Danophysa acuminata time series. As you can see, we have blooms every year with some variability. And the red dots are the DST toxin. So as you can see, they can explain up to 65% variability of the toxins, as you can see this plot. So that explains why Danophysa acuminata. The second question, why here? Because this region, it's where we have a very long time series, and it's not long, but we have several conditions. We have years of stronger blooms, years of weaker blooms, in a way that you can split into training data, that is before 2014, and test. So the machine learning model, the SBM, can learn all these patterns and also be evaluating all those patterns. So we can see actually how it's the skill of SBM in forecasting. So the first result that I have is the prediction skill improves when we use combined environment and algae input. So I have 3 different approaches here: autoregressive, environmental, and combined. In the first one, my only input is the Danophysa acuminata, the cells per liter. So which means if I want to forecast for the next week, the only information that I will use, it was the Danophysa acuminata abundance that was measured 2 days, 2 weeks ago, and 1 month ago. And here we can see that when you compare the gray line, that is the ground truth that we have, and the orange line that's forecast, the R squared is not that high. We only explain 30% of the variability in the forecast. And the SSC is the skill score compared to climatology. It's basically how much of the errors are reduced compared to climatology. When it's a negative value, it means that the climatology is better than a forecast. So here we can basically say this approach is useless because it's better to use climatology because they're using this model. But then we go to the environment prediction, now using temperature, SST, PAR, northward winds, and eastward winds. We improve the results. We have a 40% of explanation. And now skill score is positive, meaning that it's better than a climatology by 0.08. But here there is one strong limitation. We can model more or less quite well the seasonal variability, but when we have years of stronger blooms, the environment factor, the environment input cannot predict this high peak here. And one of the reasons why is because at least in this region, it doesn't matter if this year was warmer or it has or it had more winds or different variability. The environmental factors do not control the internal variability. But I have to say the environmental factors that I consider here as inputs. And the last one, when I combine the two approaches, it's when I have the better skill. I can explain 55% of the variability of the target, and the skill score compared to climatology 0.2. And again, what is important here is the year that has stronger blooms, we could forecast that. So this is for 1 week prediction, that's 7 days lead. So the question is, okay, among all those factors, what gives a skew to the forecast? So here I'm showing one method, basically it's called future importance, done by permutation. So the idea is here, we have the initial score, the 55 that it was estimated in the test data. Here in the x-axis, it's all the input of the model, so value 0 is the algae abundance in the leg 0, and the same for temperature, and then for the different legs. I take one of those inputs, I shuffle in the test data, which means that I'm mixing the data of the samples, apply the model again, and estimate the R-squared. If the R-squared had a huge reduction, it means that this feature was highly important to the models. So for example, in the first one, the R-squared is reduced by 0.2, which means that total R-squared would be 0.35 here if I shuffle this data. So the most important factors, as you can see, is the value of 0. So for example, here 7 days lead, if I want to forecast for the next week, the most important condition is the algae abundance that was estimated today. And that because it updates the current conditions of the farm. The second most important factor is sea surface temperature, like 0, like 14, and like 28, all of them. And one of the reasons I can explain why sea surface temperature is important here is because in northern Norway we have a very strong seasonal variability in temperature, varying for around 1 degree to 16 degrees, and acuminata can only grow in temperatures higher than 8 degrees. So since the temperature can control the season when the blooms can start, when we fed that into the models probably learn that, and that's why we have a nice skew here. For the remaining variables, they don't have that much significance for the forecast mode. So it's mainly algae abundance and temperature that are important here for forecasting. So I showed the results for 1 week. What happens if we increase the lead days for 7, 14, 21, and 28? As you can see, there is not that high decrease in accuracy. In the 28 days, we still have an R-squared of 47, and a score better than climatology. And of course, we can have some randomness when the models are calibrated again, because for example, here we can see the model actually improved here, but this improved so little that it's probably handled. But basically for 7 to 28 days, we at least can beat climatology in the forecasts. So, just concluding out of my work here, a very short conclusion. I'm calibrating probabilistic regression models for harmful algae blooms in Norway. What I want to show is that we can use remote sensing and model reanalysis and algae monitoring for producing health prediction models. So basically summarizing here in this figure. We can use Earth observation data, toxicology data, and machine learning. The first two here, it's usually available in the courses or worldwide available, and we can start to produce models by this approach that I just showed. And what I'm hoping here with this, my work, my thesis, that can start a harmful algae prediction here in Norway, but by using the machine learning approach, because there are some particle tracking models already being employed here, so that could be motivated to continue work and improving the models here in the region. So, well, during the PhD, what I present here it was these 3 papers, but during the PhD, it's not the only thing that you learn. You learn a lot about what didn't work, what would be nice to do, what maybe new ideas, what we need. So part of that I taught during my PhD is what I will include in future work here in Norway. So for example, one nice idea is that we can apply those models to climate projections. So for example, here is the number of detected harmful algae blooms in different warming scenarios along the Norwegian coast. So those are just initial results, so might be wrong. I'm still on development. The second one is that we really need some ocean color algorithms, regional ocean color algorithms. Not only the coast, but going to the inner fjord areas, because it's where the farms are. It's where the shellfish farms are, it's where the fish farms are. And here it's not a graph from Norway, it's a graph that I borrowed from the Yellow Sea. But you can see the reason why. When I apply ocean color algorithms developed to the open ocean, the accuracy is not that nice. So you can see here in this example we have an R squared of 21. But when we develop regional algorithms, we can have better skill. And the last one that I'm working in the following years is that I didn't mention here the details, but those models were calibrated with a very coarse spatial resolution. So here it's in northern Norway, where the farmer, one of the farms that I work in. So this larger red box here, the average of this box is the input of my model. So you can see we lose a lot of details that I could have here in temperature and the other variables. But now in the future, we have some other models with more enhanced data. So we have, for example, 800 meters resolution that is just small dots. And when you look to the coast, is this size. And even if we develop some ocean color algorithms, we can have even higher spatial resolutions here. So that definitely would enhance the predictions and forecasts. Okay, so that's all. Now it's my thanks. I have a list to do. So first, I would like to thank my advisors for helping with these whole years. Francois, Julia, Noel, helping me with learn about climate, because I got lost in questions many times to try to understand persistence, climatology, anomalies. So thanks for the help for the whole work in these years. And thanks also for the help with machine learning, because sometimes that's very complicated. Even approach, training test, and all these validation things. And even that there was a little bit of mess in the beginning because of the pandemic and doing a home office PhD, not in Norway. I think we managed to have some nice work, so thanks for that. I would also thanks for our academic staff in general here in the NASE. For example, I think sometimes annoying because I used to get every week in the office to ask for some advice. Lassie knows that. I'm always going there asking about harmful gibbons data, what are the harmful gibbons here. For the other people that I helped, especially in the first paper, for example, Aneta and Anton, some people that gave me data when I just went to the office. And everyone in general, because here it was quite easy to get in contact with people that got some help and support for developing the PhD. And also for the administrative staff, for helping with all the institute stuff and also the Norwegian stuff that I don't understand. I think it's only Christiana here, Marian's it's in the office, but thank you for all of you. Thanks also for the friends, for everyone. For example, Lilian's here, Arten's here, everyone, Heather, thanks, it was nice years. And who I'm missing? Yeah, she's there. And a special thanks to my wife, Olenka, she's for coming all the way here to Norway and freeze with me here in the cold winter. This year, so thank you very much.
So thank you for the presentation. Now we get to the really serious part here. So I have the honor to call upon the opponent, principal scientist Peter Miller.
Hi, Edson. Thank you for that very engaging presentation. So, firstly, I'd like to congratulate you on the work that you've achieved during your PhD, and I have some particular comments based on reviewing your thesis. So, I think what comes across is that you've really achieved a high level of technical skill and knowledge of the field to do what you have achieved in publishing 2 very useful papers for the field and submitted a 3rd, and that they're connected and therefore a coherent plan for improving Norway's shellfish industry. So I think the thesis itself is it's a modern approach, and I like the fact that it's focused around the papers. So unlike the thesis I wrote, how many years ago, 30 years ago, it was a big weighty tome that about 3 people have read, and most of it not published. It's focused your effort on papers that have been published and will be well read. But the thesis itself is very well organised and understandable to introduce those papers. You show a good understanding about how you're contributing to the field and what else should be done after your work in the field. So looking at the papers themselves, there's a lot of really worthwhile results and research that's in there. The first paper on the phenology, it's very thorough, comprehensive study of how blooms come and go in those northern seas. And what I particularly focused on, what I particularly liked, was that it shows, well, it hints at the connections with climate change and how some of those bloom successions are changing. Some of that might need more explaining, but you know, the data are there in your paper. So yeah, it's good that this and the subsequent paper give an idea to the fish farmers and shellfish farmers about where are good places and bad places to site their shellfish farms. So I'll come back to the second paper, but that one, yeah, that's really useful for the citing. The third paper I thought was a really well-targeted study to get a prediction system working and tested out so that you could recommend this system to a government or to a region for using. So I think that this is clearly a good contribution to the field, and I'm sure that other people will refer to that paper as the science of how prediction continues to move forward. But you know, I think it's really nice what you showed, that you need both those you need all 3 of those factors: the environmental variables going into your jug there, the environmental factors and the prediction from the model, and also the monitoring data. So I think, you know, this shows that you certainly achieved the purpose of doing a PhD, which is you're now well trained in the tools of doing research, and that you can do novel research that people want to publish and want to read, and in that way you've contributed to the scientific field. So if I now focus on some areas that I thought we could discuss. So one area that I'm particularly interested in is the satellite data on chlorophyll. So in your first paper, I think you said that the data set, the Climate Change Initiative data set that gave chlorophyll data wasn't valid for coastal regions. My view is that may have been the case when it started that project, but I feel that the more recent data sets are suitable for looking at shelf seas and coastal seas. So, do you have some comments on why you think the data are not so valid for the waters near the Norwegian coast?
So first, I think I used the version 4 or 5, the version called CCI. If I'm not wrong, there is the report made by Silva in this product, and their advice was like it was a case 1 waters biological algorithms. But anyway, at the beginning, I thought, okay, let's try to use and see. As I have in this paper, I have some matchups of validation during the development research. Again, at the beginning, I was trying to focus more on the coast, but at the beginning it was the whole sea. But when we look into those matchups in the samples that I had also in the coast, results were not good. And the main reason is that there's not many papers in satellite validation here in the Norwegian coastal shelf, but we have a lot of biooptical modelers. So seeing what is the factors controlling the optical conditions. And one example that we have from northern Norway, we have these episodic and usually common injections of fresh water from the coast that brings a lot of sedum, or colored dissolved organic matter. And this sedum, it dominates the blue to the green spectrum. So even that I know the ocean CCI is not a simple blue-green band it splits in different optical classes and use the most suitable models. If this sedum dominated at least half of the spectrum in this region, that could also explain why it didn't could match up with the surface chlorophyll. Here in Norway, it's not a huge problem, the non-algal particles contaminating the spectrum, but it seems that the sedum is the most important reason why it doesn't work very well. And the problem is, if you have sedum controlling the spectrum, which also can contaminate the chlorophyll estimation. If you look to the phenology, we can have some pulses of fresh water that might be sedum. And if you look to the phenology, we could understand that's a bloom, but it's only sedum. So that's what the reason at this moment we decided, okay, let's create a buffer zone. I think it was 30 kilometers, because it might contaminate our signal, it might bias our results, and then it's safer to work with the open ocean, remove this part, and look into the phenology. So that's why I came to this conclusion that it's better not using him in the Norwegian coast.
Okay. That's a good response, and yes, it's a good justification for your decision. But I hope that from version 4.5, with this blended approach where they look at the different water classes and give a combination of chlorophyll based on open ocean or seaborne dominated, that it should be accurate enough, I would have thought, to use near the coast. I can see that might complicate your results for the phenology, so I take your point there. But I was concerned that you seem to take from that you wouldn't use chlorophyll at all in your later papers. So how come? So I can understand that it might confuse the phenology, but why did you not use chlorophyll as an indicator?
We also thought on that because chlorophyll, it might be not that accurate, but it could give some skill in the models. The true answer is in the beginning, at least with the forecast, with third paper, we tried. It didn't give any skill. But also, I thought if I'm going to use chlorophyll only because it's an optical response and it gives some skill. Why I wouldn't use it just the reflectance, for example? Why not use reflectance on that? So I thought on that, but then if I use reflectance, it would be more monitoring than a forecasting, but also could give some skill up, because if you detect some, let's say, brown or red bloom, it could be related. But then at the moment, the idea was more for using environmental factors that can trigger the blooms than the factors that are response to blooms. For example, it changes the color, would be a response to the bloom that are already happening. So that's why I made the decision about not using chlorophyll and then not using remote sensing reflectance. And it was when I thought, okay, maybe we should think about some ocean color applications near the coast and near the fields. And then, okay, maybe I should save this idea to the future and use this data. So that was the main reason.
Okay, yeah, I think my colleagues might have some more information and consideration for that area later on. Thank you for explaining that. Now, so you said that the second paper, the one about probabilistic models, is in review. Can you give me an idea of where, like what stage is it? Is it just submitted, or is it? Have you got feedback yet?
Yes, we received comments from 2 reviewers, and it was kind of funny because the first author was really nice. He really liked the work. I mean, with few comments of improvement. The second reviewer, it seems he didn't like so much, or she didn't like so much, but the main comments were, okay, maybe it's a nice work, but doesn't fit its journal. Because it's in the environmental data science, which I disagree. I have to write the response yet, but because my work is not full biology, I'm not a biologist, it's not full machine learning, I'm not a computer science, and it's a mix between anthropology, machine learning, and that is the purpose of this journal. But one of the comments that I think it's important, I should make changes in this now, is to explain how uncertainty on the input would affect the accuracy of the models. For example, I'm using model analysis, remote sensing, but if I use data that has large errors, for example, depending on the data, the errors of estimations are larger, how that would impact accuracy. So that was a quite useful comment that I should consider now when I review.
Okay, thanks. The reason I asked that, I'm certainly not involved in the review of your paper, but if I was, I would have commented that I felt a bit confused by the story of the paper, because for the readership you're looking for, which is the shellfish industry and the shellfish farmers, associations and the science behind that. They're looking for a model that can accurately predict what's in the water now and what's going to be in the water a few days ahead, so that they can make a decision about that. There also might be decisions about, as you say, where to site future shellfish farms. But I found that there was a bit of confusion in your presentation in that paper, that the work was all about an overall probability of halves occurring in different areas in any year. It's kind of in a generic year. So you showed that there might be an increased probability that in, say, summer in this area you get more blooms of this species. But when you read the paper, it read more like used words like accuracy of prediction, and to me that gave an impression that you were saying that the model could predict that there was a bloom happening in a particular month in a particular area, but that wasn't tested. You didn't actually have data to say how well does it predict a bloom occurring now. It was, you've got data over three years, say. I think you combine data over several years, and that gives you again an overall probability.
Yes.
But that's not what the end users want. They want to know, is the bloom happening on this year, in this month? So can you comment on that possible confusion?
Yes, so it depends on the definition of the end users. I'll explain because I know, for example, there's the report of Mel from FAO. They want like a prediction, okay, in 2 weeks there will be a bloom. But this idea of seasonal probability regions where it's less likely, more likely, it came from meetings with the IMR, the Institute of Marine Research here in Norway, and some feedbacks that I had from them that they are more in touch with the farmers, it's not us, and they were interested in knowing, okay, can you say where there is which periods there is increased chances of having a had, there is a less increased periods, not for using for predictions of make like final decisions, like let's harvest all the shellfish before get contaminated. To make more complementary decisions. Okay, now we have some increase of probability, so what we should do? We should increase the monitoring, because now, for example, the toxins is every month now. But in this period that we have increased probability, should they do every week? So the idea is in this sense, it's complement and help monitoring and different mitigation options, but not to give a final decision to the farmers like stop farming or farm all you have because there will be a bloom. And I know that if you ask to the end users, the farmers, they would like to have this information because let's say it would be the most refined information, the best thing that they could have. But I have some criticism about this approach. That is, let's say, it's more of a classification, just yes or no, bloom or no bloom, do something. Because at least in the world, I have to make just some comment. For example, I think it's in the Galicia region, in Portugal, Spain, the harmful blooms are connected with some upwelling system. So when they have a rapid increase in chlorophyll, they know that our ecosystem thing will respond, and then they will probably have a harmful blooms. So then when they go to classification approach, they can have some nice results. But in the approach that I use it here, for example, using the actual conditions of temperature, salinity, fire, and mixed like that, this is not enough for making this kind of prediction because we know harmful algae depends on nutrients, but we don't have that for putting the models. Some algae depend on prey availability of very specific algae. We don't have this information to put in the models. So if you have long time series enough and we try to make classification as yes or no, we are quite good in predicting when there's no bloom, because like if it's too cold, there's no bloom, that is quite simple. But if you say there is a bloom, there will be a lot of errors. So that's why you can take some papers and some research will see, okay, the model works well, but there's many false alarms. So that's why I moved to this kind of probabilistic approach with some feedback from IMR. And with this idea, you don't have all the information for giving like a very accurate, like say in 2 weeks there will be a harmful bloom, like 100% chance. Because if I do that, I will have many false alarm hits. At least for my experience here in Norway. Again, in the regions, just one variable may be enough to give like a feedback for the whole system then using this approach. I mean, I would like to give like this accurate prediction, but at least for here in Norway, at the moment, the way that I tested, it doesn't work.
Well, an important part of my role is to give you advice on the paper that's not published yet. So I completely understand that wasn't your intention to give that kind of prediction. But when I was reading the paper, I thought it was. So that is my main reason for wanting to give you the feedback. And if it goes certainly to another journal, you might have a reviewer that also feels misled that they're going to get a prediction that would allow them to make that kind of decision about what's going to happen next month, rather than in general what's going to happen in the month of May. So yeah, I'd be very careful each time you say prediction and accuracy in that paper, that you each time that you say that it's a probabilistic prediction, it's it's not so much. I wouldn't use prediction. It's more probability. I can give a few more specific comments, but that's my recommendation for that. So let me look at what else we have here. You described that very nicely, the way that the paper is focused on worthy goals, and I agree that what you showed about the changes due to climate change and other changes would be really useful to the right people reading it. It's like another thing that you learn in your PhD, so that you're ready for the world of science, is how readily scientific results can be misinterpreted by the media and by the world out there. So I've got several examples from our group where we said things like we've got a relationship between the ocean surface mixing and acidification, and then suddenly the headline is "Satellites can see acidification in the ocean," and that these papers get attention, but for not necessarily the right reasons. It's good to get publicity, but people so much want satellites to be the tool that solves all their problems that they're on the lookout for the information. So you need to be aware of making sure you have a clear message. So let's see what else I have. I thought that this is in the thesis, which is approved already, but as a comment, I felt that that there's probably more in the literature that could have been included there on prediction and early warning of harmful algal blooms. So it seemed that it was maybe could have gone into a little bit more depth for giving a range of views on the techniques and the history of how people have tried to detect in the instructions to thesis as a whole, but that was my view. I think I've covered most of my concerns there. There's some interesting aspects that you gave at the end of your seminar there about future work. So, so in terms of climate projections of harmful algal blooms, how would you find how would you find the right readership for that kind of study? You know, how much work do you think is involved, and what kind of work would you do to to give more detail? Because people have said we think that harmful algal blooms are increasing with climate change, but do you agree with that? And how would you get, if you had more research funding to do that?
Okay.
How would you give more detail on that question?
Well, the first part that I think we're doing now is the probabilistic model that we're creating, so applying climate projection. So that's one way, because we have where the farms are located. We apply the modern climate projections, with there some distribution where it can be made between the data sets. Since it's the observed frequency, we can basically estimate how many hubs, using the farm system that we have today, how many. That is one projection, but of course we have to make some assumptions when you make this kind of projections. First, it's like the evolution of the species that can adapt to changes, and the other, it's like the background information. For example, nutrients are not taken to the model, but we have to assume the nutrients will constitute more or less the same. So I think one approach for climate projection could be using this model here. But I think for more details, we're going for more information about different approaches. I'll take some budget for making the modeling of the growth responses of the algae for some physical factors. And by physical factors, not only temperature, because I've read some works that make the modeling of the temperature of the growth response, and then applies this growth response to the climate projections, and they assume that if there is increased growth rate, because waters are warmer, there will be more hubs. I made some initial tests with using at least 15 years of data. It's not exactly true. And that's because temperature also affects the other organisms competing for the same resources, the prey, predators. So it's not exactly only the response to temperature. So what I think that's really necessary is also include salinity, because we expect the discretioning of the coastal waters. So that is important to take into account how we would impact the hubs in the coast. But also response to nutrients. Nutrients can get more complex because not only climate changing impacting the coast, it's like also human settlements, urbanizations, and many other different factors. Could be more complicated, but at least made in the modeling of growth rate responses. And especially trying to find what are the prey and grazing, because since we are working at species level, there are these selective prey. So it's not all the zooplankton grazing. We have specific zooplankton that can graze the specific harmful. So try to understand that. Because I think in the future, the main goal is to go to more mechanistic models. Machine learning can give some nice results, but there is part of understand not only the response of temperature, that it will one of the most important factors that will change in the future, but also other environmental factors and more ecological approach that involves prey and predator interactions. And then start to apply to models and see the changes.
Yeah, that's nice. Lots of sensible aspects to consider. Certainly it's not as simple as you say, the sea's getting warmer, so there will be more harmful algal creatures growing out of control. So another thing that in ocean colour we're looking towards is hyperspectral sensors. Have you come across those?
Yes, there is the Prismanel that I'm thinking using, and I'm still waiting for the dates to watch.
Yeah.
Because I think there will be like a huge step because we have more information, so we can have more information about different pigments, go to more, like, I don't think we can go to species level, like, but at least we enhance the functional types to more, let's say, what we more associate with toxic species, but yeah, I'm aware of the hyperspectrum. Maybe it definitely will be some nice work with that.
Yeah, I'm very much looking forward to PACE as well. Yeah, it's one of those things. I give these summary talks to a working group on harmful algal blooms, the ICs, and for some years I've been saying, PACE is coming, and on each talk I look at the NASA website and it gives a date, it's 18 months from now. And then I give the talk again and it's 18 months from now. But I really think that it's actually catching up now, and they're talking about launching in January, February. So, so I think it might actually be happening. Yeah, I look forward to it. But yeah, that concludes my opposition, and I will pass to my colleague back there. Thank you.
Thank you so much, Dr. Miller. Ben? Oh, you were expecting some more heat here now. I have the honor to call upon opponent, research engineer Raphael Joseph.
Okay, thank you, Edson, for this. I congratulate you for all this significant and innovative work in your field. I congratulate you also for your presentation that was really clear, and thank you for that. I appreciated also to read your manuscript that was really clear and well written. I also appreciated the format of it. It was really not easy to read, but the papers were really well. It was pleasant to read with the papers at the end, and all the methods at the beginning and so on. It was a good structure. I also appreciated the way that you presented the machine learning. I thought it really easy to read, digest, and I think that it will be maybe really helpful for other students after you, so thank you for them. Yeah, I congratulate you also for having taken in hands really diverse databases, heterogeneous and like big data like that, and to have developed such machine learning models that take time to develop, and I think that the more important things in developing such models are really to know the data that you are putting in, so we feel that you are manipulating it with ease, so I think it's a good thing. And I congratulate you also for these two papers that are accepted, and the one that is in review. I think it's a lot of work for 3 years, and there was a pandemic, so I think it was not so easy to have your thesis during this period. So congratulations. So now I have some questions about your work. First, I wanted to talk with you about the vertical distribution of this alga that you are studying, because I saw that you are using in situ data from the surface, and I was wondering what do you know about the vertical distribution, and if there are more only or more at the surface, and how it's distributing with vertical.
there is two answers. First, there is the life stages. For example, Alexandria can have cyst formation, go to the bottom and go to the surface. Usually the blooming is the surface. To be fair, at least here in Norway, we don't have that many information about vertical distribution. We have a few results in Sweden, in fjord systems. For example, the Dinophysis, usually Dinophysis acuta stay more in the surface. But acuta, Dinophysis acuta in particular, doesn't stay, the higher abundance stay like in the thin layer in the nutricon. So that gives some uncertainty because, for example, the samples that we found are collecting the first 3 meters, the nutricon sometimes is below. So the modeling could be not exactly related to the growing of the algae, but like the algae can be in dangerous levels, just down below the farm, and then there's some upwarding or something that makes it move upward. So yeah, there is some limitation about the information, but yeah, they can have some vertical distribution based on life stages, and depending on the species, they have a prevalence in the nutricon.
And so do you know how deep can be the nutricon?
I'm trying to remember now the results from the fjord, but it's definitely below 3 meters where the samples are taken.
Below, sorry?
It's below 3 meters where the samples are
yeah,
taken.
okay.
I think it's around 10 meters. I'm not 100% sure about how that is. It's not like in the deep ocean, it's really shallow, but it's not as shallow as 3 meters. I think it's between 10 and 20.
Okay.
So.
Okay. So, because, yes, to come back to this vertical distribution, so you, at the beginning of your presentation, you showed your schematic overview of the bloom with the MLD.
Yeah.
Okay, and so from this schema, we have the feeling that all is on the surface and that you have your bloom that is on the surface. So yes, you are seeing phytoplankton at the surface from satellite, but actually, for example, during summer in a lot of areas in the globe, you have the deep trophy maximum that can be productive and so it's bloom, but and sometimes the second period of bloom can be linked to this kind of vertical structures. So that's why I had this question about how it can impact, for example, the clusters that you have seen from your paper 1, and also the thing that maybe you can have toxic algae that are not seen because it's not sampled. So
how we impact the clustering?
We can.
Yeah, so well, it impacts that it's remote sense, so definitely we're only looking into the surface changes. So these 2 nice peaks that I have every year. Mostly comes from remote sensing. In my studies comes from remote sensing. We have also in situ data showing. We had some, I think it was last year it was published. There is a transect close to the Lofoten region, Northern Fjord, and it's nice the results, like it's more or less pointed based, but we can see that is what you're talking about. We have the spring bloom, but when you look at the chlorophyll A, it's going down. So it's not exactly, there is no phytoplankton. They are down. Of course, not as high as the peak of the spring bloom, but they are still there. But for the clustering, we assume that we are working the surfaces, so it's 1 divided by KD, that comes from the spectral signal. I'm trying to remember, it's only the surface, so it definitely impacted the clustering. So the cluster should be interpreted only as the surface changes. And about the subsurface,
surface or the layer that is seen from satellite. Okay, thank you. For this clustering, I had a question actually. I was wondering if you have chosen another method, for example, by clustering pixels in a different way. Do you think that you would have seen other results? So what I mean is that I know some other papers that for example, cluster things to have more dynamic clusters, you know. And so I was wondering if for this application more on climate change, what you would expect for the results if you have used another methodology.
You mean the clustering technique, like k-means,
not
or just the general method?
No, not the technique, but the way that you have clustered data.
Okay,
Like you have clustered your
I-
data pixel by pixel for all the time series, if I well understood.
Yeah, so there's two ways of that we can have a look into the relationship between climate change. I think another one that we're talking about is like having clustering the seasonal pattern and then mapping how the clusters change of the reads. So we can see the cluster moving depending on the year. That's one approach. What I used here, it was for clustering the whole time series, because I would have not only the seasonal patterns, but also where they would be changing the similar way. So for example, we can have more or less on average seasonal signal very similar, but one would be changing in one way, maybe one would be changing another way. So when we cluster the whole time series, the idea was that to capture the whole variability, and then we could have a look into it. Okay, what was this variability? We have the seasonal signal, as I focus on showing this in the map, but we also have if the signal would be changed. So that's why I did in this method, clustering the whole time series, instead of mapping the cluster, how the cluster. It's also a really nice approach.
I think it's complementary because maybe if you cluster from a seasonal perspective and you could see, for example, that you have the phenology that is changing, for example, in your northern cluster, you could see how will be the new phenology.
Yeah.
You see what I mean? And
yeah. I think it would be nice because I think we could even here in Norway see some northward shift depending on the pattern.
yeah.
Yeah.
So that's why I think it could be just you know seen in the two ways. Okay, thank you. I had a question more related to the paper 2, but it's the same for paper 3, but so I was wondering why you are not trying to retrieve directly your concentration in cells per milliliter instead of having like an absence or presence. Like you if I don't if I understood well, you take this threshold that is given from part or no from a sanitary
like a regression, right?
Yeah, okay, and so you are trying to retrieve this absence or presence, right? But you know the cells per milliliter.
Yeah, cells per liter.
Yeah, or per milliliter. No, it's per liter. Sorry, but
so why I didn't target the abundance?
yes, in a quantitative manner more than.
Okay, so when I was developing paper 2, it was more or less at the same time with paper 3. So the reason why I'm not calibrating one regression model for the whole Norwegian coast is because if you try to mix the places, the results are bad. It doesn't work. But for probability, it's more simplistic to calibrate and it works. So that is the reason why.
So you tried with the...
Yeah, if you mix the farms, let's say it's more complex to try to model the algae abundance. If you try to do that mixing the farms, the results are not that nice. So, but when working with probability, it's a less complex estimation because just like presence, absence, and it works better. So this is the main reason. Of course, I would prefer a model of algae abundance for the whole coast, but when I go to more regression and forecast approach, like trying to predict the algae abundance, I need to calibrate each model to individual farm. Not exactly individual farming because we tried, for example, the one that we calibrated, we just applied in the closest farm, that it was around 50 kilometers from there, and it worked better than I expected. So not exactly only one farm, but if you try to use all those farms, it doesn't work.
And do you think that it could be related to the distribution of your data? For example, if you have some data where you have a lot of really low abundance, or?
It's not exactly, there's a way of normalizing, make some normalizations, but the main problem, what I think it's because there is other variables that control the hubs and we are not putting that in the model. So for example, if in one farm we learn some relationship between temperature and long species of algae, if you just do some simple linear relationship, you can find something that is significant, but we are under the assumption, for example, there's nutrients there. But if you go to another farm, the nutrients configuration might be different. So when you try to apply it, there's an offset or even just no relationship. So when you start to mix out the Norwegian coast, it doesn't work. And I think that is one of the limitations with this approach, because if you need to apply to each individual farm, we need, I don't know, farms with 10 years of data. It's not all the farms that have data. So that's why I was working with probabilities, trying to implement something that it works in all the Norwegian coast and give some results that can be useful. But for the ones that have enough time series, we can do this algae modeling.
So that's why you have this paper too that is based on all farms data,
Yeah.
and your paper 3 is based on only one farm data. And so I was wondering why the...
Yeah, because if you want algae abundance, we have to narrow down to a very specific region.
And did you try with... Yeah, you tried with... Okay. So it means... So it comes to another question that I had for the... you mentioned the portability of your methods.
Yeah.
So it means that if you want to have this method available for another farms, you have to do again all your methodology and to...
Yeah, I mean, just run the codes again with a new table. So that is the idea. It's not apply the calibrated models, like the calibrated model in Northern Norway, and apply in other region. No, you run the process of training. So you need the data of armorphology in this place, and then you run the calibration again. Because you have, like, let's say, planning for each individual region. So that is the idea of extending the method. But then, at least we come here with this limitation. We have around, I don't remember now, 60 farms, at least 60 points, and only 9 that has at least 10 years of data. Then we could do something similar. Some farms are new, they only have 3 years, so we don't have many years for training and testing. Usually you see people doing cross-validation when they have short time series, but my experience with cross-validation, if you do that and they have a final test, it doesn't work that well. So I mean, the data will grow in the future, because but there's this limitation of, because it's not mechanistic, it's statistical. And statistical, it's dependent on the region it was calibrated, so that is the limitation. So that's why there's also probability for the coast.
And I was thinking also, I guess that for some areas you have to add some ancillary data, like river runoffs or something like that.
Yes, in the paper 1, paper 3, you use some river data for interpretation. In the paper 3, that it was the forecast model, it was, I'm trying to remember which variable. It was correlated with another variable, so when it wasn't temperature, I don't remember now. But when you use the river data, it didn't improve the results. Okay. And I was with the idea of only using remote sensing. Because like if I'm using river data, it's not everywhere that it has a river data. Even here in Norway. I mean, there's a lot of data here, but like the flow, it was the data that I use of the rivers in a long time series matching is not everywhere. So it could work in the paper 3. It wouldn't change the results, it would be more or less the same. So that is why I just, okay, let's use only remote sensing, because if I want to adapt to the region, I know that there will be data there.
But maybe it could help in another focus on other areas.
Maybe, yes, yeah, especially because there is no information about water stratification. That is really important in the coast. But again, it is in the inner fjords, and it should help.
Okay. Yes, I think you mentioned that that something that could improve all of this is to use regional models. Right? Regional models. To use regional models to improve.
Erosion color?
No, for your model.
For modeling.
Data,
okay. Then,
you mentioned it, no?
Yeah, yes. Basically, what I'm saying is the Northeast 800.
Because I was wondering why you did not use it directly.
2 reasons. At the beginning, I was with the idea of like being extendable, adaptable, and be using in any region. And for example, this Northeast, it's only the Norwegian coast, so it's only in Norway. And also because I got hands on this data more or less in the middle of my PhD, so it was like, okay, this data exists. I wasn't at a point that I was, okay, should I update all my research now? Or I already have all the results in published. So that's I was with the idea. I already have results. It's nice because it's using data that is almost available worldwide. But definitely in the future we should use this more enhanced data for the Norwegian coast, because then we can go to the inner fjords. Here it's only focused on the coast.
Okay.
But yeah, there was more or less a development plus the reason of trying to make something that will work everywhere.
So the advantage would have to have a better geographical resolution first, and
yes, because yes.
And also temporal. Do you have a difference on the temporal scale or
Influence on the temporal scale?
for the models that you would use?
Not exactly, because I'm limited by the audio data that is weekly. So we can have like, I think every 6 hours, I'm not sure, but again, the audio data is only weekly, so for making the matchups, it will be weekly anyway. I think the most important factor is spatial resolution, because like I'm working with a big area average, but it's not that simple, especially in the coast. You can have like fronts, we can have like stratifications, height variability that could be important, which I should test now in the future.
Yeah, so you mentioned that you could use reflectances instead of chlorophyll concentration from satellite with Peter.
Yes, if I was looking to if I have choose a biased chlorophyll that it may not work very well on the coast, I would rather use the remote sense reflectance.
Yeah, and it probably increase the performance of your model now to use it.
Yes, but there's there's the other mention that why I didn't use the remote sense reflectance. Another reason is because when I was developing these probabilistic models, the idea, okay, we have the responses from temperature, and now we can apply to seasonal prediction, like for maybe over the next year, and see how the probability will change, or dectal prediction, and like the climate projections that I was having these ideas more or less in the middle of PhD. If I had as input the remote sense reflectance, I cannot have projections on the remote sense reflectance. So that was also one of the reasons.
So you already had it in mind.
Yes.
Yeah. Okay.
And not for more or less mixing the two approaches, the paper 2 and paper 3, for making our probabilistic predictions, and then I can use the ocean color, and then use the remote sense. I mean, I can use the chlorophyll, but I can just put the remote sense reflectance and let's say the machine learning.
Yeah, so you will avoid all these problems of algorithms and.
Yeah.
Actually, I think that you will be you will go further because you will have a different wavelengths, and maybe with the species that you target, the machine learning model will.
It should be better than just using chlorophyll if I'm using as input.
Yeah.
But the interesting part of the chlorophyll is not using as input, but also the chlorophyll itself can give something about harmful high biomass blooms. I'm not sure yet about the remote sense reflectance. I still have to find some paper. I didn't find that much, but what I'm really concerned about working the coast and the fjord is like adjacent effect and atmospheric correction. So I don't know how accurate the remote sense reflectance in these regions. It's still something I have to check, but if it's everything okay, it should be good.
Okay. Yes, just to finish with this, maybe in your paper 2, it would be valuable to add, for example, the application that you see for the future to explain why you are not using this chlorophyll or reflectances data, and not just to say that there is no coastal algorithm. Maybe just to be more
I mean, you mean by the Philosophiae algorithm? Because when I say coast algorithm, I was thinking more like inner fjords and like, for example, I think the ocean CCI is 1 kilometer spatial resolution right now, right? For example, for my idea now, it's the future is more Sentinel-3 also, and then if it was solid, and then signal-to-noise ratio, the Sentinel-2.
Yeah, so maybe just to add that you don't have the geographical resolution that is enough for the scale that you are studying. But I was, I had this question of why you did not use it, because it appears really straightforward to use it for this application. So, and I was actually for this paper I also agreed with this misunderstanding. I understood reading your introduction what you were doing, and when I read the paper too, I was not sure that you are that for this story of prediction. So I agreed with Peter that maybe it's not so clear when we read the paper. But so.
There's no question about this paper too. Then it looks like they're more or less misleading, right? When I'm explaining what I do. Okay.
But it's just when we read. Because after when we see the map and you say okay, it's to give information on where they can put the farm and so on, we see the application and the thing that it's valuable, but it was more when we read it, it was not so clear. So the main purpose. And I think that Peter is right. It's maybe just vocabulary.
No, but that's good that you have this concept. I have a very long extended deadline, so I can incorporate those comments with the reviewer comments. Should be okay.
I wanted to ask you also about uncertainties that are linked to your models. If you try to derive some, or because so for now you have also you have only the, for example, the probability of occurrence, right? But not with the uncertainty from your model.
Okay. Yeah, it was. Okay, so you're asking if
I'm asking if you have tried to if you have tried to derive some uncertainties with your estimations.
Okay, for this paper to know, and again it was one of the comments of the reviewers, where it's the uncertainties.
Yeah.
But I have what it would be called the uncertainties. I didn't present here, but we have the response about input, one of the plots, and we can see that the responses are very smooth. So we can see that if the errors are in this range, the impact shouldn't be that large. So, I mean, it's the code that I have about showing uncertainty, but I still have to work something else to give the uncertainty about that. But, it shouldn't be that large because of the smoothed response of the environmental factors. There is this part of using SVM at a point when we play with one hyperparameter that's gamma. We basically define how smooth will be the response. At the beginning, I was trying to learn how to work. There was a really problem with overfit because there was a high gamma, and like the response would be like to the temperature, instead of being Gaussian, it would be like a mess. So at least the response now in the model is Gaussian, so the uncertainty if there's like a 0.5 degrees of error shouldn't be that large. But I don't have that assessed like in a statistical level to see if the correlation agrees or something like that. So yeah, but I know it's something that I should incorporate that, but I didn't have a look into.
For the paper 3, you don't have also uncertainties associated, right?
I remember it like something. I think it was doing, not in the final test, but it was doing the cross-validation because we had k-folds. Now I'm trying to remember because for some reason I think it didn't go to the paper, this part. Because we have the k-folds, all the grid search, we have the distributions of the F-square. But I think it didn't go to the paper, this part. And I think I just summarized it like we use a grid research, but yes, it wasn't to the paper, but yeah, it should be there. So.
And you mentioned the sensitivity from your inputs. And so we saw that some inputs have not a lot of sensitivity in your model. And so my question was, so does it mean that they are not so important? And if yes, if you removed it, would it have an impact in your model? I mean, is your model sensitive to inputs that are not important?
Okay, so more or less, but there is a limitation of that method that I didn't describe here in details. For example, if I take one of these inputs and shuffle, I'm losing the information. Technically, I would be losing the information of this input into the model. But if I have another input that's correlated with that, this information is just there. And it also depends on how the model learns how to use it. So it's not exactly say that it's not useful, because if I remove, it changes the model. But the changes are not that large. That's why I just continue with the final data, especially because PAR had some skills in longer forecasts. I have some skills, but there's this limitation about the method, because it's correlated with the other variables, which are, especially when we have time lags, because then we have the autocorrelation input, then there is this limitation. So not exactly they are not important, they might be just correlated with another feature that it wasn't shuffled.
Okay.
So that is one. But it's hard to completely say it's not, it is important, because like, for example, PAR showed that there is no importance, but we know that PAR is important for algae. Even though it seems that it's not for the model not learn, it just reads correlate, because it's correlation. So,
Yeah, I think it's not so easy to...
yeah, it's easy to separate it.
Yeah.
But I think it's the closest that we have. We have other feature importance methods. Instead of shuffle one, it's shuffle all of them except one. But then there is more different uncertainties and stuff. And it's hard to understand.
Yeah.
But yeah, there's some limitations in methods that I cannot exactly tell 100%.
And so the SVM is less performant if you have... no, it's not less performant if you have correlation between variables. I mean, if you put too much inputs, it won't be less performant?
Not exactly. SVM works quite well with a lot of inputs. I know there are some, I think, for example, handle forest may not work very well if you have more features than variables. But usually SVM, its concept idea is to work with hyper dimensions. So it works well with many different features. For example, if you think about just a polynomial model of one feature, let's say temperature. If it's at 2 degrees, we could have 2 features, and I can work with almost infinite number of features. But this is one skill of SVM, working with as many features needed. Not only is the original input, but it is converts to the future space, and where it is compute the hyperplane. So usually it's known for working with many features. So wouldn't be that hard problem.
I don't know how much time I...
It's a little bit up to you.
Since when I am talking?
Half an hour.
Ah, okay. Time flies. Okay, maybe just one last discussion. It was... yeah. For your paper 1, so for the cluster, then you linked it, so you have shown this figure with the onset of the bloom that is linked with... yeah, it was SST.
With the correlations?
Yeah. And so, yes, I was wondering if you have also studied other criterion, like, for example, the duration of the bloom, because you mentioned the onset of the bloom and the magnitude, no?
Yes.
Yeah, and so you said, for example, that a deeper MLD will have a bloom that will occur before, and also will be more intense.
Yeah, when I have a shallower mixed MLD, yeah.
Shallower.
Yeah. Yeah, I didn't have time to present more details, but especially in the Barents Sea opening and in the beginning of the Norwegian Sea, when we have I think the strongest spring bloom. I don't remember the year exactly, but the shallowing of the mixed lake depth starts more or less beginning of April. So beginning of April start to shallowing, it become very shallow in the April. And this year the spring bloom just start early and go really fast, and it also finishes earlier and go really fast. If you take and there is another year that it was end of April, almost May, that it start shallowing, the blooms basically doesn't develop. I mean, develop very weakly.
So it's more an indication of your stratification here.
Yeah, it's an indication of the stratification.
Not the MLD that goes. I thought that it was, for example, the MLD that is going deeper during the winter. So, for example, if you have a really deep mixing event, you will have more nutrients that are brings, and then maybe you will have a bloom that is more intense.
Yeah, no, but the correlation is with mixed lake depth in the spring. Of course, we need the deep mixed lake depths in the winter to be more nutrients, because if you don't have that, you have less nutrients, and that would be also an impact. I didn't have a look into that, but it would be really nice, because we can have some variability in the winter. But when I showed this result, it was the spring bloom is only the mixed lake depth during the spring.
Yeah, okay, so it's at a T0.
Yeah, because shallow mixed lake depth during the winter would be the opposite effect.
Yeah.
Because it would be less nutrients. So that is important to say during the day spring.
Okay, so maybe it would be interesting to. And yeah, also I was wondering if you have studied other criteria like the duration of the bloom, because you have the intensity, but sometimes you have a bloom really intense that decreases really rapidly, and then you have not so much biomass if you integrate it all the time. You can have a bloom that have a long duration.
More like look into the primary production, net primary production.
Yeah, or just biomass.
Yeah, I mean, if it's a weaker bloom but very long, you probably would have different primary production, maybe more than one, very shorter and but stronger. I didn't have a look into this because at the moment I was really focused with the blooms, the intensity of the blooms, the start of the blooms, so very into the phenology of the blooms. But that would be something that would be interesting to see, which one would be looking into the primary production. And I had focused on the blooms, I want to continue.
Yeah, but not in terms of primary production, but just to say that sometimes you can have more in time, you know. You can have, if you have a persistent bloom, it will be more biomass during the...
You mean after when it reaches the peaks, how long it stays, right?
Yeah.
Yeah, that I didn't assess what happens after the bloom. So it was only exactly between onset, peak, duration. And here by the duration, I use the definition between onset and peak, because I know some bloom phenology studies, it's also considered after the peak, but that I didn't have a look at. It was only this bloom when you have like the rise of biomass.
Thank you.
Thank you so much, Dr. Souza. Then, are there any questions from members of the audience? Yoni is considering, but he's giving it a pass, I see. So, it doesn't seem to be the case. So, then the committee will withdraw for a few minutes. All you guys will be waiting here, and we will come back with our conclusion. Please.
Yes, not
à la fin, quand tout le monde part.
Ça dépend du temps de la déviation.
That's on you, if you need oxygen behind.
And up and down.
Fully open that.
Just do it like this.
I think it's enough like that, right?
Yeah. Let me see.
Well done so far.
J'étais le premier.
C'était une expérience.
J'étais le premier. J'étais le premier truc. Il y avait
I'm just.
Do you have to write something? Or is it only...
What's
Do you have to write something?
that afterwards.
Like a little note or...
No, I think they're doing,
Oh, no, okay.
they're doing it afterwards. You just have to sign the paper. No, it's just. I guess they have.
So, the committee has now evaluated the performance of the candidate, and I'm happy to conclude that the committee has accepted both the presentation and the defense, and will thus recommend that Edson, Filisbino, Freire da Silva should be awarded the degree of Philosophia Doctor at the University of Bergen. The University Board will be notified accordingly. These thanks already as part of your presentation, but here I'm not actually at part number 9 of my program. The full dissertation has come to an end and the candidate may have the floor.
I didn't know.
But now you can do it as a doctor.
Okay, so. Well, I think I will repeat myself again. I think you're okay, it's my defense, I can just listen for minutes. Just following what I said earlier about my thanks for my advisors. I know, man. All these years. Also thank for the opponents. I know that it's a lot of work when we receive a document that it has inclusion method limitations. Thanks for accepting, for evaluating this work. I really like it that you accepted because if I evaluated to my doctoral degree, I might have some really nice experts to approve me. So thank you very much for our evaluation, for coming here and with the discussion, and with the nice suggestions that I will definitely incorporate. So thank you very much. The table. Thank you very much for guiding here. Here, I think I just. Thank you for everyone. I think for the announcement, for us here, I have to say it's a really nice environment to do research. And I had some experience. We students talk with each other to say it's really nice when we can not only communicate with advisors, communicate with other research, other experts, just go to the room and just talk and like do research. So that is really a nice support about anything here. So thank you very much.
Procedure has come to an end, and we enter in academic procession and we will leave in academic procession. But as a recognition of your new, let's say, dignity, you will lead us out, and I believe there are some bubbles and cake facing us on the other side of the door. So the committee, if you can join Dr. Silva. Okay. You have all the time in the world that you're
You should walk this way. So.
So thank you. See you in 3 meters.